Hello students, this is Dr. Faizan Mirza. In this video, I'm discussing Feb March variant of this MCQ paper 2023. So in this paper, the first question came for magnification. You are provided with this photomicrograph here. Neutrophil is shown to you. The diameter is being taken from the line P and Q. They're asking you to calculate the actual diameter. So we know actual to calculate actual, we have to calculate image upon magnification. This is the formula. So how do we calculate the value of I here? So whenever this is the condition, we measure the distance between the line provided to us. We measure it in millimeter. After measuring it in millimeter, we multiply it with 1,000. So we get the answer in micrometer. Once the answer is in micrometer, we divide it by magnification. So applying this formula, we get the actual diameter. So this comes out to be the only option for the answer which is viable. Question number two, a student calibrated an eyepiece reticule using a stage micrometer. Each division of the stage micrometer is 0.01 millimeter. With a 10x magnification objective lens, 10 eyepiece gratical units meshed 10 divisions on the stage micrometer. So this is the 1:1 ratio in which they basically are there. So 10 eyepiece gratical units match with 10 divisions of stage micrometer. Furthermore, we know that the distance is 0.01 millimeter between each division. So these two bullets actually mean that one gratical unit is equals to 0.01 millimeter. This is the calibration that is provided to us. The same microscope was used with a 40x lens instead of 10 magnification object lens to measure the diameter of an alveolus. The diameter of the alveolus is found to be 96 gratical units. The eyepiece reticule was not changed. The best estimate for the diameter of alveolus is, now what we will do, we know the scale, one graticle unit is equal to 0.01 millimeter. We know this, we'll calculate it for the 96 graticle units. You just cross multiply, you get the value of x. X is by multiplying 96 by 0.01. We get the answer of 0.96 millimeter. We multiply it by 1,000 to get the answer in micrometer. So it's 960 micrometer. Now, at 10x, 960 micrometer will be 10 times magnified. So this 960 micrometer will become 9600 because this is 10 times magnified. Now, if it is changed to 40, this eyepiece is changed. Sorry, the objective is changed to 40. We'll apply the formula of this magnification. Again, this actual is equals to image upon magnification. The diameter that we got was 9600. We'll divide by the magnification 40 and we get the answer to 40 micrometer. Which statements are correct for chloroplast and or for mitochondria? All the options provided to us are correct except for ATS ribosomes because they have 70S ribosomes. The electron micrograph of some root cells are shown. If it's root cell, the root cell cannot have chloroplasts. So which cell structure are not usually in from the root because chloroplasts are the only in the mesophyll cells, which are palisade and spongy, or the guard cell. Otherwise, chloroplasts are not present. Dialysis disc tubing is an artificial partially membrane with a pore size of approximately 2.5 nanometer. Glucose molecules have a diameter of 1.5 nanometer, which means they can pass through. The pores, what else can pass through, right? Bacteria, no bacteria are much larger. Hemoglobin is a large protein. Hemoglobin and ribosome, again, we know the diameter is 25 to 18 nanometer for ATS and 70S ribosome. Fructose will be of the similar diameter as glucose because they are just the isomers of one another. So yes, the answer is 4 only. What is present in all the viruses? The viruses have either DNA or RNA. So all viruses cannot have ribose or not have deoxyribose. Yes, adenine can be present because that's a nitrogenous base common to DNA, RNA both. Thymine also cannot be the answer. To estimate the concentration of glucose in an unknown solution, equal volumes of a range of known concentrations of glucose per test tube. Now, if this is a range of glucose known concentrations, it means this is their independent variable of glucose, which were each mixed with the same excess volume of the Benedict solution. After mixing, the solution were placed in a thermostatically controlled water bath at 90 degrees Celsius for 3 minutes. The unknown solution was then treated in the same way and the colors of known and unknown were compared. What is the independent variable? What were you changing? You were changing the glucose concentration here. So concentration of glucose in this case will become our independent variable because independent variable is the one that we actually change ourselves during the course of an experiment. And when we are changing this, the dependent variable also changes. And when the dependent variable changes, our experiment results actually are varying because we are constantly changing our independent variable along the experiments. Question 8. Which statements are correct for amylose and for amylopectin? We can recall the structures of amylose and amylopectin. All options except 3 are correct. They are carbohydrate molecules. They are formed by condensation reaction. They contain 1,4-alpha glycosidic link. They are linear molecules. No, they are not. This is helical and this is branched. So that's an easy question. Which statement is correct comparison between saturated triglycerides? Saturated triglyceride will have carbon-carbon double bond. It will be a triglyceride molecule, something that I've drawn here. An unsaturated triglyceride molecule, unsaturated should have a kink somewhere because of the double bond in one of the tails, approximately the same molecular mass. So how can they have the same molecular mass? Unsaturated triglycerides have more double bonds and fewer hydrogen atoms than saturated. Yes, that's true because when there are more carbon-carbon double bond, there will be less hydrogen. There, unsaturated triglycerides have fewer double bond. That's absolutely incorrect. Unsaturated triglycerides have more double bond. Yes, this is correct, and more hydrogen. No, that's not possible. It is contradictory. Unsaturated triglycerides have fewer double bonds. Again, this is wrong science because it will have more double bond. That's why it's unsaturated. Question 10. Part A, B, C, and D are labeled. Which is the correct label for the description? D is phosphate group. So this is the correct label. C is not the hydrophobic and the glycerol. B is not the hydrophobic end of the triglyceride molecule. And this is not a triglyceride molecule. It's a phospholipid. That's why it's not triglyceride. That's why option B is incorrect. Fatty acid and hydrophilic end of molecule is A. Again, that's incorrect because this is the region which is hydrophilic. Which row correctly shows the level of protein structure that can be held together by each type of interactions? We know hydrogen bonds are present in secondary structure. So yes, secondary structure and tertiary structure can both be the answer. Hydrophobic interactions, tertiary is good. Covalent, we know peptide and disulfide are the only two type of covalent bonds present, and they are present in the primary, tertiary, as well as quaternary structure if the quaternary structure is there. So the answer that comes out to be will be same. Which molecules contain at least three double bonds? Saturated triglyceride molecule will have three fatty acid tails, each having a C double bond O. Collagen will have these polypeptide chains. Again, C double bond O is present because of the carboxyl end. The same goes for the hemoglobin as well. So the answer is D because they both are proteins. They have this carboxyl end, and the triglyceride will have three carboxyl end as well. Which diagram correctly shows hydrogen bonding between two water molecules? So we know that the hydrogen bonding should always be there between oxygen and hydrogen, and they both should be sharing a partial charge, in which the partial negative charge should be on oxygen and the partial positive charge should be on hydrogen. Otherwise, all other options are incorrect. Question 14. Which statement describes an example of an extracellular enzyme? It is the enzyme which is released outside the cell. So other than A, there can be no other answer because all these enzymes, they work within the cell. Which row is correct for the enzyme that catalyzes the reaction by lock and key hypothesis? Lock and key hypothesis or induced fit hypothesis, both means the enzyme actually lower the activation energy, they won't raise the activation energy. This description is for induced fit, and this description is for the lock and key hypothesis, with which the enzyme and substrate are complementary fit for one another. So the answer will be B. A scientist investigated the rate of breakdown of hydrogen peroxide. Four experiments were carried out with all these conditions which are told to you. The results are sketched on the graph. You can just see the rate of breakdown of hydrogen peroxide and substances provided to us. This line shows that it's constantly rising and it stays low. So it means that the rate of breakdown of hydrogen peroxide was very low as substrate concentration was increased because the enzyme was not there. That's why substrates were being broken down to products grab a bit very slowly. But when you add enzyme substrate together, the reaction rate of fission is very fast initially, and eventually it becomes saturated because all the enzymes have saturated and the reaction reaches Vmax. This will be the Vmax region. Anyhow, second and third shows competitive and non-competitive inhibitors. That's why the Vmax change, the stays same, and here the Vmax changes. So this is the graph that we can recall from the enzyme chapter. Now they're asking about which is the substrate only, which is the competitive inhibitors. 4 and 2 will be the answer here as per the annotation that I've added according to the explanation that I just provided. Question 17. Which of these substances can pass directly through the cell surface membrane using a carry without using, which means that this is allowing simple diffusion to occur. So we know this is a charge, this is large, so they can't move by simple diffusion. Whereas this is a gas, it can move by simple diffusion, so 2 is the only option. This 3 is glucose, it is a polar molecule, so it's no sorry, it's not, yeah, the C6H12O6 is the glucose, so it will need, it's polar, it would need a channel or a carrier protein. What happens to the cell surface, to the surface area to volume ratio of a cube when the length of each side is doubled? So it all decreases by half because when you increase it, the surface to volume ratio becomes halved. So that's a fact, and that again comes as a B. Which events are part of mitotic cell cycle? So in mitotic cell cycle, all these phases are there, 1, 2, and 3. Question 20. The estimated number of RBCs in the human body is this 2.5 to 10 to the power 13. It is estimated each day 2.5 10 to the power 11 RBCs are removed from circulation, so they must be replaced from the bone marrow. Which percentage of the total number of red blood cells is replaced each day? Now, what will we do? RBC replace upon total RBC. Total will be our denominator. RBC replace will be our numerator. We'll multiply it by 100. You perform this simple calculation. You get the answer 0.01. Multiply by 100 and you get 1%. DNA polymerase catalyzes the condensation reaction between molecules during semiconductor replication of DNA. What is joined by DNA polymerase? A nucleotide is joined to another nucleotide. Two polynucleotide strands make up a DNA molecule. Which description is correct? The percentage of cytosine is the same as guanine. Yes, because they are complementary bases. A pretty straightforward question with all three options, which can be outright be rejected. 23. This gene, PDX1, actually is a protein, is found in a wide range of animal species where it is regarded as essential for normal metabolism. In sand rats, the gene code PDX1 protein has a much lower proportion of AT nucleotide than other organisms. So if it's having less ANT nucleotides, so adenine, thymine, we know have double hydrogen bonds, and CG have a triple hydrogen bond. So there will be more CG in this gene. Finding the complete DNA sequence of this gene in sand rats has been difficult. Sequencing involves splitting of nucleotide one at a time at a single at one time, one at a time from single-stranded DNA. What could account for difficulty in finding DNA sequences of PDX1 gene in sand rats? PDX1 gene is present only in small proportion in the nucleus. No, there's no such evidence provided to us why it can be difficult, why this particular gene sequencing can be difficult. This fact, this statement doesn't align with the information provided. The PDX1 gene is transcribed only in some cells. Again, we are not given this information. The strength of hydrogen bond between two strands of PDX gene is unusually high. Yes, because of more CG bonds. So option 3 is correct. Different tissues in a plant were supplied with a radioactively labeled substances to identify which tissues were actively synthesizing mRNA. So mRNA is being synthesized by wherever uracil is present, ribose is present, because we know that these both will be used. Adenine inorganic phosphate alone will not be used. Okay, so we move on to question 25. Which row correctly matches the structure and function of phloem sieve tube elements? Structure: peripheral cytoplasm with no nucleus, provide little resistance to flow possible. This is for sieve elements because this is how the sieve tubes function. A straightforward question. Which feature of xylem vessel elements helps adhesion during transpiration? So adhesion is between the walls and water molecules. So if the tube is narrow, it would allow more water to cling to the walls. Lignin form a secondary wall. Well, secondary wall is made by cellulose only, and then lignification occurs. The new vessels carry extra water. No, we have not studied. There is no certain fact which can be related to adhesion as far as new vessels are concerned. There are no cross walls. Yes, this is correct. But this has got nothing to do with addition, because addition is all about the property of water clinging to the walls of the vessel. Mass flow is bulk flow movement of materials from one place to another. How many vessels listed carry fluids by mass flow? So all these carry materials from one place to another. So all four of them will basically be there in the answer. Cellulose, lignin, and suberin are components of various plant cell walls. Description of these cell wall components are listed. A component of Casparian strip, we know it's suberin. Redirect water into symblastic pathway, Casparian strip, suberin again. Hydrophobic components of cell wall, lignin. Water interact with this molecule for the apoplastic pathway. So yes, this should be cellulose. So which will correctly match to the three descriptions provided? The answer will be D. If you annotate it properly, it's not a difficult question. The arrows show direction of water movement across the plant root from water to the soil in the xylem. So this direction is shown to you. So this is the symplastic pathway we know. The plasmodesmas are being crossed. The cell membrane is being crossed. Whereas the second arrow, which is following just the cell walls, this is the apoplastic pathway. So 2 and 4 are the ones which are apoplastic. 6 cannot be apoplastic because it's entering into the cell. Which row correctly identifies source and sinks for source and sink? So sink should be root cell, which is absorbing. Yeah, sink should be that because this is where this being actually moving. Storage cells of seeds that is starting to grow. So a seed which is starting to grow, it will have certain storage cells which will be providing glucose to the cells that are growing. So these storage cells will become the source. So that's why the answer is A. A very close option could be sink if you do not read the question correctly. If instead of storage cell, they just give you seeds that is growing, you would consider seed to be a sink because it's growing. But no, they have asked you about the storage cells present inside the seed, which are starting to grow. The photomicrograph shows a section through a tissue of an artery, which will correctly describe the type of artery, whether the blood inside the artery is oxygenated, deoxygenated. Now, this is a kind of a tricky question. You need to identify what this diagram is. So this diagram shows to you a lot of alveoli, and these alveoli, we know they allow oxygenation to occur. And this vessel here, it's having a thick muscular wall. So this must be a muscular artery. Now, if it's a muscular artery, the muscular artery must be coming from the pulmonary artery and taking the deoxygenated blood close to the alveoli. So eventually the blood becomes oxygenated. That's why the option A is correct. Heart surgery may cause a decrease in the transmission of impulses from in the Purkinje tissue to the right side of the heart. What is the possible effect of this decrease? Impulses would be delayed? No, that's generally the normal course. There won't be any additional effect there. The muscle of the right ventricle would contract slightly more slowly. Yes, because the transmission is delayed towards the right side. This is the correct answer. The most muscles of the right atrium would not contract fully. Now, contraction being fully or being not fully is completely a muscular mechanism. It has not got to do much to the impulse being transmitted. The sinoatrial node would transmit fewer impulses. No, SA node is the pacemaker. Here we are discussing only the Purkinje tissue. 33. The diagram shows a network of blood vessels supply blood to the muscles in the human system. X and Y are the two regions provided to you. What is correct for comparison between the blood at X and the blood at Y? So at blood, we know the blood pressure is high. So the answer should be either C or D. At Y, we know that the water potential of blood compared to the water pressure, the blood should be low. So should be high because this is still not the venule end. So that's why if it was given by here, so in that case, it would be lower. So in this region, it would still be high. So that's why the answer stays C. Scientists have not shown that scientists have shown that oxygen dissociation curves for hemoglobin of smaller mammals are actually to the right of those of large mammals. So if it's this is the normal dissociation curve, it shifts towards right. It goes towards this side. So whenever there is a right shift, it means that it's easy for hemoglobin to be unloaded from easier for the oxygen to be unloaded from hemoglobin. And if the normal curve is there and there is a left shift occurring, in this case, the hemoglobin losing oxygen getting unloaded from hemoglobin, it will become more difficult. In this case, they are saying it's a right shift, so unloading of oxygen becomes easier. Option C is correct. What is the effect of increasing the concentration of CO2 in blood? So if you increase CO2 in blood, chloride move out of red blood cell? No, they move in. Increased concentration of hemoglobinic acid? Yes, HbA increase because H plus ions increase, then they bind to hemoglobin. Decreased concentration of hydrocarbonate ion in plasma? No, HCO3 are not there and actually are not affected by this because HCO3 is in the plasma are in the red blood inside the red blood cell cytoplasm and not in the plasma. Decreased concentration of carbamide in hemoglobin? No, it would increase as well because CO2 will bind to hemoglobin to form carbamide in hemoglobin. Two requirements of an efficient gas exchange system are listed for surface area and for short distance, which will correctly describe how alveoli are adapted to meet these requirements. So we have to restrict ourselves for this answer to the gas exchange system and the alveoli and not go to the cardiovascular changes associated with the adaptations. So the large surface area of the alveoli is completely because alveoli are folded and are interconnected. And the short distance here is because the walls of alveoli are only one south. The other options do not comply with gas exchange and the alveoli, rather they are the adaptation of the cardiovascular system. What defines the infectious disease? Symptoms are caused by a pathogen that is transferred from one host to another. Yes, a generalized statement. Other options actually are too restricted, so we can't use those as an answer. A hurricane destroys a large town on the island. People move away from the town and set up tents where sanitation is poor. So if sanitation is poor, that means the transmission of cholera will be high because within its life, they will be exposed to using water which is not which might be contaminated in that region. Plus, we know that disasters which allow destructions to occur on land, they lead to more cholera outbreaks. Question 39. Which statement correctly explains why viruses are unaffected by penicillin? So that's a fact-based question again. So just recall it basically disturbs the peptidoglycan synthesis. So option D is correct. Which processes characterize the mode of action of phagocytes? Phagocytes do not produce antibody. Yes, receptor binding is there, endocytosis occur, hydrolysis occur as well, exocytosis also occur of the debris. So yes, 2, 3, 4, 5 all are correct. So with this, I wrap up this video. I hope this video was helpful for you. I would recommend that you try to attempt this paper all by yourself and see if this video worked out for you. That's it from my side. Thank you so much.